So, good afternoon. Welcome everyone to this tutorial on metabolic pathway modeling using ordinary differential equations. My name is Veronica. I am a PhD student in the group of Luis Serrano. And although my main field of study is transcription, today we will be talking about metabolism. So the tutorial today is going to be divided in four parts. The first one will be about how we represent our biological knowledge in the form of a system of ODEs to make these models. The second part is about how we use experimental data to fit the parameters of these models. The third part is about analyzing the steady state and what happens when we make small perturbations to this steady state. So it's a stability analysis. And the fourth part is about more practical application of these models. It's about metabolic control analysis and identifying those key points in the pathway that can be targets for engineering. So if you remember Jonathan's talk on Sunday, he was describing that when we are doing some modeling, we have to find a certain compromise in between the size of the system or the scope of the model that we are working with and the level of detail we can achieve. So in this case, this is a different representation of the same. On one side, we have qualitative models such as networks that can be very big, represent very big systems in biology, but with very little detail. On the opposite side, we have kinetic models. These kinetic models have a lot of detail. We can have ODEs or also stochastic models, but they're limited to very small systems. So today we're going to focus on these ones. So what do we do to represent a metabolic pathway in the form of a set of differential equations? So we're going to start with this toy example in which we have a very small pathway with some reactions. For example, there's some production of metabolite A. A is producing both B and C. C is degraded, but it's also used to produce D, and D is degraded. So we can make a list of all the reactions occurring in this pathway that we have here, and then how we convert these reactions into the set of differential equations. So we can do this by, as Professor Bas was explaining this morning, writing a differential equation for each of the chemical species in our system. And these differential equations are going to be defined by the producing reactions minus the consuming reactions for each of the chemical species. Once we have all the differential equations written, what we have to do is to replace these reactions by the appropriate rate laws for each of them. To do this, we need to know a number of things regarding the biology of the system. We need to know the reaction stoichiometries. For example, here is one molecule of A producing one of B or one of C and one of C or not. We need to know also whether the reactions are reversible or not. We need to know about the kinetics of the reaction. Are they Michaelis-Menten, which is usually the case but not always, and how are they regulated? And also we need to know the kinetic constants for each of the reactions. So we're going to go through all of this now. So first of all, I'm going to make a short review on Michaelis-Menten kinetics. I don't know whether you see this well or not, but enzymes work forming a complex in between the enzyme and the substrate, which is this ES complex. And then once we have this complex formed, the catalysis of the enzymatic reaction happens and the products can be released. So with this, we can assume that the speed of the reaction, that is the rate the rate of production of the product, is this catalytic constant multiplied by the concentration of this complex, the ES complex. So So then we're going to make one assumption, that is that this ES complex is going to be very constant and low. Constant and very low concentration. So this differential equation that represents the rate of change of the ES complex, we can equal it to 0. And maybe it's better that I write it because I don't know if you see it. This is forward. This is going to be equal to 0 because we're assuming that this is constant. So we can reorganize this, this part of the equation, and we get that this ES concentration is going to be equal to once we have this, we can convert all of this in the Km, so that the inverse of Km actually, so that this is so now we're going to make another assumption, that is that the total enzyme concentration, so the one that's free and the one that's forming this complex, is going to be constant. And we can make this assumption because the time that it takes to produce new enzyme in transcription and translation is going to be much longer than the time it takes to complete enzymatic reactions. So once we assume this, the total enzyme is this, we can replace in this equation that we had before, so that we get to the following. So now we have to replace this in the first equation, the one that we were using for the actual rate of the reaction, the product production. So once we change this, we know that this is equal to k cat and we can actually replace this. We are going to say that this is going to be the maximum rate, so we're going to call this V max, and this is how we derived the Michaelis-Menten equation, which is written here. I guess you can see it in your slides. So we can have some modifications on this equation given by inhibitors. We can have inhibitors that are small molecules that bind to the same place as the substrate of the reaction. And then we have competitive inhibition, and this is the equation representing it. We can have some inhibitors that bind only when the substrate has already bound the enzyme. They only bind to the ES complex. Then we're calling this uncompetitive inhibition, and that's the equation that represents it. And finally, we can have some inhibitors that can bind either the enzyme alone or the ES complex. And then we call this non-competitive inhibition. So this was the basics for irreversible kinetics. But now what happens in biology is that most of the reactions that we're going to be working with are reversible. So when we have reversible kinetics, we have this. So before we had the same equation, but this didn't exist. Now we also have this. So we're going to call this K1, K minus 1, K2, and K minus 2. So we have to modify these equations in order to have the same Michaelis-Menten equation for the reversible kinetics. So in this case, what we have for this case is that we have to add a term. So here I'm going to use the new notation. This is K1. This is K minus 1. And this is K2. And we have an extra term because we are also producing this complex via this reaction. So it's K minus 2 multiplied by the enzyme and the product. So with this, we can derive a Michaelis-Menten equation for reversible kinetics, which would be this one. So in this case, we have the forward speed of reaction, the forward rate, multiplied by the substrate, divided by the constant for the substrate, minus the reverse, the rate of the reverse reaction, multiplied by the product, divided by the kinetic constant for the product. And here we have the same. So what we can do now is that, assuming that we are in equilibrium. In equilibrium, the speed of the rate of the forward reaction and the rate of the reverse reaction are the same. So we can make this equal to 0. Once we make this equal to 0, we get rid of this part. So all of this is equal to 0. We're organizing this a little bit. We can get to the Halden relationship, in which we have the equilibrium constant, which is equal to the concentration of the product in the equilibrium divided by the substrate concentration in the equilibrium. Or this is the same as forward rate multiplied by this kinetic constant for the product divided by the reverse rate and the kinetic constant for the substrate. So reorganizing this equation that we have here with this equilibrium constant, we can get to this. This is more interesting because we are only measuring this forward reaction speed. We don't usually measure this reverse one. And we can introduce this equilibrium constant to replace this. So we can have different mechanisms of reaction other than Michaelis-Menten. We can have, or modifying Michaelis-Menten, we can have allosteric regulation, we can have ordered mechanisms, we can have constant fluxes, etc. But I'm not going to go into this in very detail today. So during the rest of the tutorial, we're going to work with a model of the central carbon metabolism in E. coli. This model was published in 2002 by Christophe Chassagnol and collaborators. And it includes the reactions regarding the glucose import, glycolysis, the pentose phosphate pathway, and some biosynthetic reactions. So in total, the model accounts for 18 species, 48 reactions, like 30 of them are kinetic rates. We also have dilution equations, and one equation that is a bit special for the extracellular glucose. And apart from this, we have 7 other functions that are not coupled to the system of ODEs that describe the changes of the co-metabolites like ATP, AMP, ADP, or NADH, NAD, etc. that describe in an empirical manner the changes of these metabolites. So in the model, E. coli cells are growing in a chemostat. A chemostat is a type of bioreactor in which we have a closed system that is well mixed. We have stirring inside, and we have a feed that is supplying fresh medium with all the nutrients that the cells require, and we have an effluent. And in the effluent, we have all the metabolites that are in the medium, also cells that are flowing away of the chemostat. So we have and one of the advantages of this type of systems is that E. coli can grow, or any bacteria can grow in a physiological steady state. So we can get a constant growth, and this is very useful to use in modeling. So this, I hope you see it better in your slides, is the representation of all the pathways included in the model described in this paper. So with squares, we have the enzymes, we have in circles regulatory effects of different metabolites over some reactions, and we also have indicated which of the reactions are reversible or irreversible. So, as I mentioned before, we can describe the system by a set of differential equations. Each of them is referring to one single chemical species, one metabolite of the model. And here we have the producing and the consuming reactions, as well as the dilution. In this system, as you can see, many of the reactions are Michaelis-Menten, some of them with one substrate, with two, most of them reversible, some of them irreversible. But as I said, in biology, this is the most usual thing to encounter. And these are the 7 empirical equations describing the changes of ATP, ADP, AMP, etc. So these metabolites are not going to be coupled to the model. So they're going to be treated a bit specially. We will see this in the exercises. So the first exercise, which I think for this one you don't need actually to program anything, is to take some of the reactions happening in this network and define what are the rate equations of these reactions. So I chose 5 of the reactions in the model. I'm describing which are their kinetics, whether they have activators or inhibitors. And with this and the slides that you have before, you should be able to describe which are the kinetic rate equations for each of them. So the idea is that I can give you, let's say, 5 to 10 minutes, and then I will write the solutions in the whiteboard. If anybody wants, what you can do is, once you have the equations written on a piece of paper, you can go to this script in the materials that you have in the website and replace these chunks of code by working code representing these equations. But I think for now I'm happy if we're able to write this in a piece of paper. We're on the side of the plot in which we can have very detailed models, but they're very limited in scope, so they have to be small, because, and one of the reasons of this is that we have a lot of unknown parameters that we should estimate to be able to work with this kind of models. So here we have only one of the kinetic rates of the model. And only for this one equation, we have up to 8 parameters to estimate, 8 different parameters. In the entire model, we have up to 116 parameters, and we only have 18 mass balance equations and 18 metabolites that we're modeling. So we need experimental data to fit our model. So why do we need to address this parameter estimation problem? First of all, we need to know the dynamics of the system that we are studying. And this is the set of kinetic equations that we were writing before. These are the ODEs. Then we need a cost function to optimize. So the goal that we have now in this part of the tutorial is to find a set of parameters that minimizes this cost function. So what is this cost function? We can have different choices for it. So this cost function, what it's going to do is compare the results of my simulation with the experimental data that I'm getting from the laboratory, and trying to minimize the difference in between the values I'm getting from my simulation and the experimental values. For this, we can have different types of cost functions, like generalized least squares or likelihood. So basically what we do is, like in both cases, is for each experiment, we have different observables, but for the different observables, which are the different species that we're measuring, we can have different samples because we're measuring at different time points. And for each sample or each different time point, we have to measure what is the difference in between the measured data from the experiment and the simulated data with the set of parameters that I'm choosing. So we make the difference, we square it, and we multiply it by some factor. In this other case, with log likelihood, we have the same case. We're trying to measure the difference in between the real data and the simulated one, but instead of normalizing by some parameter, we're dividing it by the variance of the data. So if we're measuring the data and we have different replicates, we can know what is the variance and we can use it and put it as an input in our cost function. We will be using this log likelihood cost function in the tutorial later. So we need to know what is the set of parameters we want to estimate. We have these 116 parameters, and it is very useful if we can have some initial guess of what are the values for these parameters. And for example, this can be done with a literature search. So we can find in the literature some parameters from other experiments found for these parameters we want to estimate, and we can start our search with these values. We need to know the initial conditions of our dynamics, but if we don't know them, we can include them among the parameters to be estimated. And another thing we need to know is the upper and lower bounds of the parameters. So we can also constrain the search space for our parameters if we know something about the biology of the reactions occurring. So if we have the minimum and maximum possible values for this, we can constrain a lot our search, because if not, the bounds are minus and plus infinite. And finally, we need a solver, which is an algorithm that is going to simulate our system of differential equations and try to find a set of parameters that minimizes the objective function. There are different algorithms. Some of them are global optimization. Others are doing local optimization. So the difference among them is that, imagine that I have a very simple example in which I only have one parameter to estimate. And this parameter, this is the value of the cost function, and this is the value of my parameter. If I'm using one local algorithm to optimize this function, I may start at this point. I may continue searching. Here I found a minimum, but it's not the global minimum. So if I'm using a local algorithm, here it says that I'm going up again, and it will indicate that this is the minimum that optimizes my cost function. But this is not true. I have another point here that is much better to fit my data. So There are other algorithms that use this local search, but instead of starting at one point, they use multiple points to increase the chances that I'm finding this global minimum. And there's a third class of algorithms, the ones that we're going to use today, that they try to scan the whole possible space to find the actual global minimum. So some examples of this kind of algorithms are scatter searches, simulated annealings, genetic algorithms, and there are many others. We were running the simulation for a while without any perturbation, and when we were plotting the results, we had only straight lines. This is because we were in steady state and the system, the concentrations of the different metabolites were not changing. So when a system is in steady state and we make a small perturbation of this state, like the glucose pulse, what happens to our system? So does it go back to the same steady state situation, meaning that this is a stable steady state, or we have changed to another point, etc. So for example, imagine we have a pendulum. In a pendulum, we can have two different types of steady state. If we have the pendulum down here, this is the axis and this is a rigid rod. If I move the pendulum a bit to the right, it will bounce a little bit, but it will come back to the same steady state situation. However, if I have the pendulum up here, exactly in equilibrium here, if I move it a little bit towards one side or another, it will come here and it will get to a new steady state, but not the same one as before. So this can happen also in the steady state that we're finding in these kind of models. But of course, there are also other types of steady states. So to explain this, I'm going to use a very well-studied model, which is the Lotka-Volterra for studying ecosystems of prey and predators. So in this model, we're going to have the prey represented as U and the predator as V. We're going to have the birth rate of the prey represented by alpha. So the rate of change of the prey is going to be alpha U. And the death rate of the predator is going to be represented by beta, so it's going to be here. Gamma is going to be the fraction of prey that's caught per predator per unit time. So, and E is going to be the rate of biomass conversion from prey to new predators. So prey is going to be eaten by the predators, and it's going to turn into new predators, as it says here, that they're going to die somehow. And the prey is going to be born, and they're not going to die unless they're eaten by the predators. So in order to analyze what happens to the system, first we need to determine which are the steady state points. So if we make these rates unchanged, equal to 0, we can find out that there are two possible solutions for this system. First, that we have no prey or predator, which is a trivial solution. There's nobody in the ecosystem. And second, that we have some numbers of predator and prey. And in this case, for the system to be in steady state, these are the values for the different populations. So we have two steady state points for this system, and we're going to study whether they're stable or not. So to study the stability of a system that is close to steady state, for which we make a small perturbation of the steady state, we have to calculate the Jacobian of the system. And the Jacobian, it's the derivative of each of the differential equations of the system as a function of each of the variables. So in the system, we have two variables, which are U and V. So we're going to calculate the derivative. Sorry, better here. This is the differential equation of U. So we're going to calculate the derivative of this one with respect to the prey. we're going to calculate the derivative u with respect to the predator, and we're going to do the same with the other differential equation with respect to the prey and with respect to the predator. And this is what we call the Jacobian of the system. So once we calculate these derivatives, this is what we get. These are the values that we get for them. And what we do is, for this steady state point that u is equal to 0 and v is equal to 0, we replace here, and this is what we get. So to study the stability, we need to know the eigenvalues of this Jacobian matrix. So these eigenvalues are going to tell us what is the type of this steady state. And the eigenvalues of a 2 by 2 matrix are very easy to calculate by hand. So they are the solutions of the determinant equation, lambda multiplied by the identity matrix minus this Jacobian. So we have this here. So this lambda multiplied by the identity is nothing else than minus this, the result of the Jacobian and the steady state, and the determinant of this we have to equal to 0. So we calculate so the result of the determinant is lambda minus alpha multiplied by lambda plus beta. This has to be equal to 0. So the result, the eigenvalues of this matrix are alpha and minus beta. So there are different types of equilibrium for systems of two equations, and we can know in which type we are according to these eigenvalues. So in our case, we have two eigenvalues, one larger than 0 and one smaller than 0, because it's minus beta. So in this case, we are here. We have a saddle point. This means that if we are moving in one direction from the equilibrium, which is here, if I move exactly in this direction, I'm going to go back to the same equilibrium point. But if I move in any other direction, which is not this straight line, if I go, for example, here, I'm going to go away from the equilibrium. So we can do the same for the other steady state point, which is alpha divided by gamma and beta divided by E gamma. We do exactly the same. We calculate the Jacobian, and we replace this by the values in the steady state. From here, we calculate which are the eigenvalues. And in this case, we have this result, which means that we have some complex numbers here. So in this case, we are in a orbit because we have one positive and one negative, which means that we're going to be here. So in this plot, I'm showing you, like, in the parameter, well, no, this is a phase plane plot, which means it represents the different possible concentrations or numbers of prey and predator. So we have the two steady state points. Here we have the saddle node, which is when we have no prey or predator. So in this case, if we move across this line, we have more predator, but there is no prey, so they will die, and we will come back here. However, if there is some prey, some predator will appear to convert this prey into predator biomass. So we will move here, and we will have in this kind of orbits. And in this point, we are in the other steady state point. So in this point, we have this kind of equilibrium in between the prey and the predator. But as long as we move a bit outside it, we're going to have orbits around this equilibrium point. Identify which are the key reactions of our system that are good candidates to be targets for engineering. Because it tries to find which are the parts of the system that have larger control on the fluxes and the species concentrations. So, to be very brief about this, we're not going to do the exercise, but a flux is the rate of turnover of molecules through a metabolic pathway. So in this case, we have a linear pathway with a source and a sink with different intermediate steps. So the flux would be like the rate at which molecules are going through this pathway. So what we want to do with metabolic control analysis is to identify which are the reactions that have a largest influence in this flux, which are the reactions that if I increase the rate, I'm going to increase the flux over the whole pathway. This is very interesting because imagine that in this central carbon metabolism pathway, imagine that I want to maximize the production of pyruvate. I can calculate which are the reactions that have the largest control on the pyruvate concentration and try to overexpress the enzymes that are responsible for increasing the flux towards pyruvate concentration. But I'm running out of time, so I invite you to read the slides and do the exercise 4. I will provide the solutions for this, and if you have any question to this respect, I will be happy to answer, but I'm sure that there's Buzz over there in the end who will be much better than me in answering questions. So with this, I'm going to finish by just summarizing that we've seen how to define the equations of an ODE model of metabolism. We've done the parameter fitting of this ODE model by using experimental data. And finally, we've performed stability analysis, studying how the system behaves when we are close to the steady state with some small perturbations. So here you have some useful reading in the topic. And this is what I wanted to tell you today, so thank you for your attention.